Hey guys, tonight we are diving deeper than the height of Mount Everest, but in reverse, plunging into the crushing darkness of the ocean's deepest trenches, where the water pressure would instantly turn your body into something resembling a deflated balloon. You definitely wouldn't survive this. We are exploring one of the most mind-bending mysteries of our planet. How creatures not only survive but thrive under pressures that would obliterate any surface dweller in milliseconds. So before you get comfortable, take a moment to like the video and subscribe, but only if you genuinely enjoy what I do here. Drop a comment letting me know where you are listening from and what time it is in your corner of the world. Now dim the lights, maybe turn on a fan for that soft background hum, and let's ease into tonight's journey together. You begin your descent from the sunlit surface waters, where familiar dolphins and sea turtles glide effortlessly through their liquid world. The pressure here feels like a gentle embrace, one atmosphere pressing evenly against every square inch of your imaginary diving suit. But as you sink deeper, that gentle embrace begins to tighten its grip with mathematical precision. Every 10 meters down adds another atmosphere of pressure, and you are heading toward depths where that number climbs into the hundreds. At 100 meters below the surface, you notice the first changes in the marine life around you. The colorful tropical fish from the shallows have vanished, replaced by creatures with larger eyes and more streamlined bodies. The pressure here is already 11 times greater than what you experience at sea level, enough to compress a beach ball to the size of a marble. Yet the fish swimming past you seem completely unbothered, their movements fluid and natural, as if they were floating in air rather than liquid concrete. You continue your descent into the twilight zone, where the last traces of sunlight fade into an eternal blue-black darkness. Here, at depths of 500 meters, the pressure reaches 50 atmospheres. To put this in perspective, imagine 50 cars stacked on top of every square inch of your body, and you begin to understand the crushing reality these creatures call home. The water itself becomes denser, more viscous, creating an environment so alien that it might as well be another planet entirely. The creatures you encounter here display their first obvious adaptations to this high-pressure world. Their bodies appear almost gelatinous, lacking the rigid structures that surface fish depend on for survival. Jellyfish pulse through the water with ethereal grace, their translucent bells contracting and expanding in slow hypnotic rhythms. These are not the delicate, fragile creatures you might expect to find in such a harsh environment, but rather perfectly engineered biological machines, designed to thrive where others would perish. As you pass the 1,000-meter mark, entering what scientists call the bathypelagic zone, the pressure climbs to over 100 atmospheres. Here lives the anglerfish, that nightmare creature with its bioluminescent lure dangling in front of razor-sharp teeth. But despite its fearsome appearance, the anglerfish represents one of nature's most elegant solutions to extreme pressure survival. Its body contains no rigid bones, no air-filled organs, nothing that could be crushed or compromised by the immense weight of water above. Researchers still argue that we have barely scratched the surface of understanding how these pressure adaptations actually work at the molecular level. The deeper you go, the more mysterious these survival strategies become. At 3,000 meters, where the pressure reaches 300 atmospheres, you encounter creatures that seem to defy the basic laws of physics and biology. The giant tube worms that cluster around hydrothermal vents at these depths present a particular puzzle. These alien-looking creatures, reaching lengths of up to 8 feet, have no digestive system, no mouth, no obvious way of obtaining nutrients from their environment. Instead, they rely on symbiotic bacteria living within their tissues to convert chemicals from the vents into usable energy. But even this remarkable adaptation pales in comparison to their ability to withstand pressures that would instantly crush any surface-dwelling creature into a dense, lifeless mass. You drift past these biological impossibilities, watching them sway gently in the deep ocean currents, their red plume-like structures filtering chemicals from the water with mechanical precision. Each one represents millions of years of evolutionary trial and error, countless generations of creatures that gradually adapted to survive in conditions that seem fundamentally incompatible with life itself. The water around you has become something entirely different from the liquid you know at the surface. Under such extreme pressure, its molecular structure changes, becoming almost solid in its density and resistance to movement. Yet somehow, the creatures here navigate through this liquid concrete as easily as birds fly through air, their movements betraying no sign of struggle against the crushing forces that surround them on all sides. You focus your attention on the microscopic level now, peering into the cellular machinery that makes survival at these crushing depths possible. Inside every deep-sea creature swimming past you, an intricate molecular ballet unfolds, one that would make the most sophisticated origami artist weep with envy. The proteins that form the building blocks of life itself must fold and twist into shapes that can withstand pressures equivalent to having the entire weight of the atmosphere compressed into a space the size of your thumb. At the surface, proteins fold into their functional shapes through a delicate process that relies on precise temperature and pressure conditions. Change those conditions even slightly, and the proteins unfold like wet tissue paper, losing their ability to perform the chemical reactions that keep life running. Yet here in the abyss, where pressure readings climb into ranges that would liquefy most organic compounds, these same proteins not only maintain the structure, but actually seem to prefer these crushing conditions. The secret lies in what scientists call piezophilic adaptations, modifications that allow proteins to function optimally under extreme pressure. You witness this molecular engineering at work as enzymes inside a deep-sea fish continue their chemical choreography, breaking down nutrients and building cellular components with the same efficiency as their surface-dwelling cousins, despite operating under conditions that should theoretically make such reactions impossible. These pressure-adapted proteins have evolved unique amino acid sequences that create more compact, tightly packed structures. Where surface proteins might have loose, flexible regions that allow them to bend and twist during their function, deep-sea proteins are built like molecular fortresses, with every component locked into place by additional chemical bonds that can withstand the crushing forces around them. The temperature factor adds another layer of complexity to this molecular puzzle. Deep ocean waters hover just above freezing, creating a double challenge for these protein machines. They must not only resist crushing but also function efficiently in near-freezing temperatures that would slow most biological reactions to a crawl. Evolution has responded by creating proteins with enhanced flexibility in specific regions, while maintaining overall structural integrity under pressure. You observe a deep-sea bacterium as it processes chemical compounds from a nearby hydrothermal vent, its enzymatic machinery working at temperatures that would cook most surface organisms, while simultaneously withstanding pressures that would flatten a submarine. The proteins inside this microscopic organism represent millions of years of evolutionary fine-tuning, each molecular component perfectly positioned to maintain function under these extreme conditions. Researchers still argue that the mechanisms behind pressure-resistant protein folding could revolutionize biotechnology applications on the surface. Understanding how these molecules maintain their structure under extreme conditions might lead to the development of industrial enzymes that can function in high-pressure manufacturing processes or pharmaceutical compounds that remain stable under conditions that would destroy conventional medications. The polyunsaturated fatty acids in deep-sea creature cell membranes provide another fascinating example of molecular adaptation. These specialized lipids remain fluid and flexible at crushing depths and near freezing temperatures, maintaining the cellular boundaries that would otherwise become rigid and brittle under such conditions. It is like having biological antifreeze combined with molecular shock absorbers, all wrapped into the same cellular component. You drift closer to examine the gill structures of a deep-sea fish, watching as specialized proteins transport oxygen from the water into the bloodstream. These transport proteins have evolved unique conformational changes that actually improve their oxygen binding efficiency under high pressure, turning what should be a survival disadvantage into an evolutionary edge. The fish extracts more oxygen per breath than its surface relatives, despite operating in an environment with lower oxygen concentrations. The heat shock proteins found in deep-sea organisms represent another molecular mystery that continues to puzzle researchers. These cellular repair mechanisms, normally activated only during times of extreme stress in surface organisms, run continuously in deep-sea creatures, constantly maintaining and repairing protein structures that would otherwise be damaged by the crushing pressure environment. Some deep-sea proteins exhibit what scientists call pressure-induced stabilization, where the crushing forces actually help maintain the protein's functional shape rather than destroying it. This counterintuitive phenomenon suggests that these molecules evolved not just to tolerate extreme pressure, but to actually require it for proper function, creating biologic systems that would fail if brought to the surface where pressure levels drop to what we consider normal. The molecular chaperones that assist in protein folding in deep-sea organisms work through mechanisms that remain largely mysterious to surface-based researchers. These helper proteins guide the folding process under conditions that should prevent proper protein formation entirely, yet somehow manage to produce perfectly functional molecular machines that outperform their surface counterparts in efficiency and durability. You watch as a deep-sea organism repairs damaged proteins in real time, its molecular machinery working with clockwork precision, despite operating under conditions that would instantly destroy the cellular infrastructure of any surface-dwelling creature. Each repair represents a triumph of evolutionary engineering over seemingly impossible physical constraints. You witness now one of the ocean's most dramatic and puzzling phenomena as researchers attempt to bring a deep-sea creature to the surface for study. The ascent begins slowly, the specimen contained within a specially designed pressure chamber that maintains the crushing conditions of its natural habitat. But as the chamber rises through the water column, you observe the delicate balance between life and death that defines existence in the pressure gradient of our oceans. The barotrauma that occurs when deep-sea creatures are rapidly decompressed resembles a biological explosion in slow motion. You watch as gas-filled organs expand beyond their structural limits. Cell membranes rupture from the inside out, and proteins unfold like biological origami, being hastily undone by invisible hands. The creature that moments before swam gracefully through liquid concrete now becomes a victim of the very pressure differential that once protected it from surface predators. Yet not all deep-sea creatures suffer this fate, and herein lies one of marine biology's most perplexing mysteries. Some species possess the remarkable ability to survive the journey between pressure worlds, their bodies somehow adapting in real time to the changing conditions as they rise through thousands of meters of water column. These biological shapeshifters represent evolution's most sophisticated solution to the pressure survival problem. The swim bladders of deep-sea fish present a particular puzzle in this pressure transition mystery. These gas-filled organs, which help control buoyancy in surface fish, should theoretically expand and explode during rapid ascent from the deep ocean. Instead, some deep-sea species possess specialized valves and muscular controls that can rapidly adjust gas volume to accommodate changing pressure conditions, like having built-in biological pressure regulators. You observe a deep-sea fish as it begins its ascent, its swim bladder contracting and expanding in precise rhythms that match the decreasing pressure around it. The muscular walls of the organ squeeze and release with mechanical precision, maintaining internal pressure equilibrium through what can only be described as biological engineering of the highest order. The fact that this occurs automatically, without conscious control from the fish, makes the adaptation even more remarkable. The cellular mechanisms that prevent decompression damage in these ascending creatures involve rapid changes in membrane permeability and protein structure. As pressure decreases, specialized transport proteins in cell membranes adjust their configuration to maintain proper ion gradients and prevent cellular rupture. It is like watching biological levees reinforce themselves in real time as floodwaters recede. Researchers still argue that the speed of these pressure adaptations challenges our understanding of how quickly biological systems can respond to environmental changes. The molecular rearrangements required to survive rapid decompression should take hours or days to complete. Yet some deep-sea creatures accomplish these changes in minutes as they rise through the water column. The blood chemistry of pressure-tolerant species provides another piece of this decompression puzzle. These creatures maintain specialized proteins that can rapidly bind and release dissolved gases as pressure changes, preventing the formation of gas bubbles in the bloodstream that would cause fatal embolisms. Their circulatory systems function like sophisticated decompression chambers, automatically adjusting to prevent the bends that would kill human divers under similar circumstances. You notice that many deep-sea creatures that can survive pressure transitions possess highly flexible skeletal structures that can compress and expand without breaking. Their bones contain more cartilaginous components than their surface relatives, creating biological shock absorbers that can accommodate rapid volume changes during ascent or descent. These creatures essentially have built-in pressure suits made of living tissue. The nervous systems of pressure-adaptable deep-sea creatures show remarkable resilience to the electrical disruptions that typically accompany rapid decompression, while surface creatures would experience neural chaos as pressure changes affect the transmission of electrical signals. These deep-sea specialists maintain normal brain and nervous system function throughout their journey between pressure zones. Some species exhibit what scientists call pressure memory, where previous exposure to pressure changes somehow prepares their biological systems for future transitions. These creatures seem to remember the molecular configurations needed for survival at different depths, rapidly switching between pressure-adapted and surface-adapted states as conditions require. The reproductive strategies of pressure-tolerant species add another layer of complexity to this survival mystery. Some deep-sea creatures must travel to different pressure zones to mate or spawn, requiring their reproductive organs to function normally across a range of crushing to minimal pressure conditions. Evolution has somehow solved the engineering challenge of creating biological systems that remain fertile across pressure differentials that would destroy most living tissue. You observe a remarkable scene as a deep-sea creature completes its ascent to surface waters, its body having successfully navigated pressure changes equivalent to traveling from the bottom of the deepest ocean trench to the peak of the highest mountain. The fact that any living organism can survive such extreme environmental transitions represents one of biology's most impressive achievements, a testament to the incredible adaptability of life itself when given millions of years to perfect its survival strategies. You examine now the architectural marvels that deep-sea creatures use to build their bodies, structures that must withstand forces capable of crushing steel submarines like empty soda cans. The shells and skeletons you observe down here represent some of the most sophisticated engineering solutions in the natural world, biological constructions that make human architecture look primitive by comparison. The calcium carbonate shells of deep-sea mollusks present a particularly fascinating example of pressure-resistant construction. Unlike their shallow water relatives, whose shells can be delicate and ornate, these deep ocean architects built with thick reinforced walls that distribute crushing forces across curved surfaces, designed to deflect rather than resist the enormous pressures surrounding them. Each shell represents thousands of layers of crystalline structure, laid down with the precision of a master mason working in microscopic dimensions. You observe a deep-sea nautilus as it adds another layer to its spiral shell, the creature's specialized organs secreting calcium and carbonate ions in carefully controlled ratios that create materials stronger than many industrial composites. The mathematics of its spiral construction follow precise geometric principles that distribute stress evenly throughout the structure, preventing catastrophic failure points that would doom the creature under crushing pressure. The bone structure of deep-sea fish reveals even more remarkable adaptations to extreme pressure environments. Rather than the hollow, lightweight bones found in surface fish, these creatures possess dense, solid skeletal elements with internal architectures resembling the steel framework of skyscrapers. Microscopic examination reveals intricate lattice patterns within the bone matrix, creating structures that can compress under pressure without breaking, while maintaining their essential support functions. These pressure-adapted bones contain specialized cells called osteocytes that continuously monitor stress levels and make real-time adjustments to bone density and structure. When pressure increases, these cellular architects deposit additional calcium phosphate in strategic locations, reinforcing the skeleton like living buttresses that respond automatically to changing load conditions. The speed of these adjustments rivals the response time of electronic pressure sensors. The cartilaginous skeletons of deep-sea sharks represent a different approach to pressure-resistant construction entirely. Rather than building rigid structures that resist crushing forces, these creatures have evolved flexible frameworks that bend and compress under pressure while maintaining their essential mechanical properties. Their cartilage contains specialized proteins that remain elastic under conditions that would make surface cartilage brittle and prone to fracture. You watch as a deep-sea shark navigates through crushing pressures that would collapse its skeleton if it were made of conventional bone, yet its cartilaginous framework flexes and adapts to each pressure variation with fluid grace. The entire skeleton functions like a sophisticated suspension system, absorbing and distributing forces that would otherwise propagate through the creature's body and cause internal damage. Researchers still argue that the growth patterns of deep-sea skeletal structures challenge conventional theories of biomineralization under extreme pressure. These creatures must somehow precipitate calcium compounds from seawater and organize them into complex structures, while operating under crushing conditions that should theoretically prevent such precise chemical processes from occurring. The microscopic architecture of deep seashell construction reveals hierarchical organization that spans multiple scales, from molecular to macroscopic. At the smallest level, protein matrices provide templates for crystal formation, guiding the precipitation of calcium carbonate into specific orientations that maximize strength. These protein scaffolds themselves must maintain their structure under crushing pressure, creating a biological construction system that operates under conditions no human engineer has ever attempted to replicate. The growth rings visible in deep seashells tell stories of survival that span decades or even centuries under crushing pressure. Each ring represents a year of successful pressure resistance, a testament to the remarkable durability of these biological construction materials. Some deep-sea clams show growth rings indicating lifespans of over 400 years, their shells maintaining structural integrity throughout centuries of constant crushing force. The repair mechanisms that maintain deep-sea skeletal structures present another engineering marvel worth contemplating. When damage occurs, specialized cells migrate to the site and begin rebuilding using the same precise molecular techniques that created the original structure. These biological repair crews work under crushing pressure conditions that would prevent most chemical processes from functioning normally, yet they successfully restore full structural integrity to damaged shells and bones. The external shells of deep-sea crustaceans combine flexibility with strength through layered construction techniques that resemble advanced composite materials. Multiple layers of different materials, each optimized for specific mechanical properties, work together to create protective armor that can withstand crushing forces while maintaining the flexibility needed for movement and growth. You notice how these creatures must periodically shed and rebuild their entire exoskeletons as they grow, a process that temporarily leaves them vulnerable to crushing pressures until new shells harden. The timing and coordination required for successful molting under extreme pressure conditions represents one of biology's most dangerous and remarkable balancing acts, where miscalculation means instant death, but success enables continued growth and survival. You peer deeper into the cellular architecture of these pressure survivors, focusing now on the microscopic mechanisms that prevent their most basic biological components from collapsing inward like deflated balloons. The cell membranes you observe here represent some of the most sophisticated boundary systems in nature, biological barriers that must maintain precise selective permeability while withstanding forces that would instantly rupture any surface cell. The phospholipid bilayers that form these cellular boundaries contain specialized fatty acid compositions, unlike anything found in surface organisms. These membrane lipids remain fluid and functional under crushing pressure through molecular modifications that would make a biochemist's head spin with their elegant complexity. Each membrane molecule has been fine-tuned by evolution to resist compression while maintaining the flexibility needed for essential cellular transport processes. You watch as transport proteins embedded within these membranes continue their molecular shuttle service, moving nutrients into cells and waste products out, all while operating under pressure conditions that should theoretically lock these protein machines in rigid, non-functional configurations. The fact that cellular metabolism continues normally under such extreme conditions represents one of biology's most impressive engineering achievements. The osmotic pressure regulation in deep sea cells presents a particularly intricate balancing act that would challenge the most sophisticated chemical engineering systems. These cells must maintain proper internal water content despite being surrounded by seawater under crushing external pressure that constantly threatens to force excess water across the cell membrane. Evolution has responded by creating molecular pumps and channels that work with mechanical precision to maintain cellular water balance. The specialized aquaporin channels that control water transport in deep sea cell membranes operate through mechanisms that remain largely mysterious to researchers. These protein tunnels can rapidly open and close to regulate water flow, preventing cellular swelling or dehydration despite dramatic pressure variations that occur as creatures move through different depth zones. It is like having microscopic plumbing systems with automated pressure relief valves built into every cell. You observe the remarkable efficiency of ion transport across these pressure-adapted membranes, watching as sodium, potassium, and calcium ions move through specialized channels that maintain electrical gradients essential for nerve and muscle function. The fact that these electrochemical processes continue to operate normally under crushing pressure demonstrates the incredible sophistication of deep-sea cellular engineering. The membrane-bound organelles within these cells face their own unique pressure challenges, requiring specialized adaptations to maintain their internal environments while surrounded by cytoplasm under extreme pressure. Mitochondria, the cellular powerhouses that produce energy through chemical reactions, must somehow maintain the delicate membrane structures needed for efficient energy production, despite operating under conditions that would collapse these organelles and surface organisms. Researchers still argue that the speed at which deep-sea cell membranes can adjust their permeability in response to pressure changes challenges our understanding of membrane biophysics. These rapid adjustments occur through molecular mechanisms that should theoretically require more time to complete than the pressure changes themselves. Yet somehow these cellular boundaries respond almost instantaneously to environmental variations. The endoplasmic reticulum networks within deep sea cells show remarkable structural modifications that allow protein synthesis and processing to continue under crushing pressure. These internal membrane systems must maintain their complex folded architectures while facilitating the movement of newly synthesized proteins through cellular compartments, all under conditions that would cause conventional cellular machinery to cease functioning entirely. You notice how the nuclear membranes of deep-sea organisms maintain their integrity while protecting the genetic material inside from pressure-induced damage. The double membrane structure surrounding each cell nucleus must remain selectively permeable to allow normal gene expression and DNA replication, while preventing the crushing external pressure from disrupting these essential genetic processes. The vesicle transport systems that move materials between cellular compartments operate with remarkable efficiency despite the extreme pressure environment. These microscopic cargo containers must maintain their structural integrity while traveling through cytoplasm that has the consistency of liquid concrete, delivering their molecular payloads to precise destinations within the cell architecture. The cellular cytoskeleton networks that provide structural support and enable cellular movement show unique adaptations to pressure that involve specialized protein filaments capable of bearing enormous loads while maintaining flexibility. These biological cables and struts create internal frameworks that prevent cellular collapse while allowing the dynamic rearrangements needed for cellular division, growth, and repair processes. You observe the process of cellular division occurring under crushing pressure, watching as a deep-sea organism creates two new cells from one through a complex choreography of membrane formation and organelle distribution. The fact that such delicate biological processes can occur successfully under conditions that would instantly destroy surface cells demonstrates the remarkable adaptability of life when given evolutionary time to perfect its survival strategies. The waste removal systems that transport cellular debris and toxic compounds out of deep-sea cells operate through specialized membrane transport mechanisms that function efficiently under extreme pressure. These biological garbage collection systems prevent the accumulation of harmful substances that would otherwise poison the cell, maintaining cellular health despite operating in an environment that poses constant challenges to normal biological processes. You encounter now one of the deep ocean's most perplexing biological paradoxes as you observe creatures with gas-filled organs that should theoretically be impossible at these crushing depths. The swim bladders of deep-sea fish present a mechanical impossibility that somehow functions with perfect efficiency, like watching balloons remain inflated under the weight of entire mountains pressing down from above. The physics of gas compression under extreme pressure follows predictable mathematical laws that should make gas-filled organs in deep-sea creatures either explode during ascent or collapse completely during descent. Yet you observe fish swimming gracefully through crushing depths while maintaining precise buoyancy control through these seemingly impossible biological balloons that contain actual gas at pressures that would turn surface gases into liquid or solid states. The specialized cells lining these deep-sea swim bladders possess the remarkable ability to secrete gases directly into the bladder cavity despite operating under pressure conditions that should prevent gas formation entirely. These gas-secreting cells work like biological pressure pumps, concentrating oxygen and nitrogen from the surrounding blood and forcing these gases into the swim bladder against crushing external pressure that would resist such accumulation. You watch as a deep-sea fish adjusts its buoyancy in real time, its swim bladder expanding and contracting with mechanical precision as the creature rises and falls through the water column. The muscular control systems that regulate bladder volume operate through neural pathways that somehow account for the extreme pressure differentials encountered at various depths, creating automatic buoyancy compensation that would impress aerospace engineers. The gas composition within deep-sea swim bladders differs significantly from atmospheric air, containing specialized gas mixtures that remain stable under extreme pressure conditions. Some deep-sea species concentrate gases to pressures exceeding 1,000 atmospheres within their swim bladders, creating internal environments more extreme than the crushing depths surrounding them. Yet these organs continue to function as effective buoyancy control devices. The blood supply systems that service deep-sea swim bladders represent marvels of biological engineering that transport dissolved gases through circulatory networks operating under crushing pressure. specialized blood vessels called retia mirabilia form complex coiled structures that concentrate gases through countercurrent flow mechanisms, creating biological gas pumps that can generate enormous internal pressures within the swim bladder. Researchers still argue that the energy requirements for maintaining gas-filled organs under extreme pressure should exceed the metabolic capabilities of deep-sea creatures, yet these animals somehow manage to power their biological gas compression systems while maintaining all other life processes under conditions that challenge the fundamental principles of bioenergetics. The structural integrity of swim bladder walls under extreme pressure requires specialized tissue compositions that can withstand internal gas pressures while preventing gas leakage into surrounding body cavities. These biological pressure vessels possess wall thicknesses and material properties that rival industrial gas storage systems, yet they grow and develop through normal biological processes under crushing environmental conditions. You observe other gas-filled structures in deep-sea organisms that present similar mechanical paradoxes, including gas-filled chambers in the shells of deep-sea cephalopods and gas bubbles within the tissues of certain deep-sea invertebrates. Each of these biological gas containers represents a unique solution to the problem of maintaining gaseous structures under pressure conditions that should eliminate all gases from biological systems. The evolutionary origins of these pressure-resistant gas organs remain largely mysterious, as the intermediate forms between surface gas organs and deep-sea versions would presumably be non-functional death traps that could not support survival during the evolutionary transition. The fact that these sophisticated pressure-resistant systems exist suggests evolutionary pathways that operate through mechanisms not yet understood by marine biologists. The communication systems used by some deep-sea creatures involve gas-filled sound production organs that generate acoustic signals under crushing pressure conditions. These biological sound generators must create and manipulate gas bubbles for sound production while operating in environments where gas bubbles should not be able to exist, creating underwater voices that somehow function despite physical impossibility. The digestive systems of certain deep-sea fish contain gas-filled chambers that aid in processing food under extreme pressure conditions. These digestive gas organs must maintain their internal gas pressure while processing nutrients and dealing with the mechanical challenges of digestion under crushing external forces, creating biological chemical reactors that operate under conditions no industrial engineer has ever attempted to replicate. You notice how some deep-sea creatures use their gas-filled organs for purposes beyond buoyancy control, employing these structures as pressure sensors that provide information about depth changes and pressure variations in their environment. These biological barometers function with precision that rivals electronic pressure measurement devices, yet they operate through mechanisms that remain largely mysterious to human researchers. The maintenance and repair of gas-filled organs in deep-sea creatures requires specialized biological processes that can service these structures while maintaining their internal pressure and preventing catastrophic decompression. The cellular mechanisms involved in maintaining these organs represent some of the most sophisticated pressure engineering found anywhere in the biological world. You delve now into the biochemical mysteries that allow life processes to continue at crushing depths where normal chemical reactions should grind to a halt like clockwork mechanisms frozen in time. The enzyme systems operating within deep-sea creatures represent some of nature's most sophisticated chemical catalysts, molecular machines that somehow accelerate biochemical reactions under pressure conditions that should inhibit or completely prevent such processes from occurring. The metabolic pathways you observe in deep-sea organisms follow the same fundamental chemical principles as their surface counterparts, yet they operate with enhanced efficiency under crushing pressure that would destroy the delicate molecular interactions required for normal metabolism. These biological chemical factories process nutrients, generate energy, and eliminate waste products while functioning under conditions that would shut down any comparable industrial chemical process. The respiratory enzymes that enable deep-sea creatures to extract oxygen from seawater and deliver it to their tissues work through mechanisms that actually benefit from extreme pressure rather than being hindered by it. These pressure-enhanced enzymes demonstrate improved binding efficiency and reaction rates under crushing conditions, suggesting that evolution has not merely adapted to extreme pressure, but has learned to harness it as a metabolic advantage. You watch as the cellular powerhouses within deep-sea organisms continue their energy production processes, converting nutrients into usable chemical energy through reaction cascades that should theoretically be impossible under such extreme pressure conditions. The mitochondrial enzymes responsible for energy production show remarkable stability and efficiency improvements when operating under crushing pressure, creating biological power plants that outperform their surface equivalents. The protein synthesis machinery within deep-sea cells continues to manufacture new proteins at rates comparable to surface organisms, despite operating under pressure conditions that should disrupt the delicate interactions between ribosomes, transfer RNA, and messenger RNA molecules. These biological protein factories maintain their precision assembly processes, while surrounded by crushing forces that would destroy similar mechanical systems designed by human engineers. The DNA replication and repair systems in deep-sea organisms function with remarkable accuracy under pressure conditions that should cause errors in genetic copying processes. The enzymes responsible for maintaining genetic integrity show enhanced stability under crushing pressure, ensuring that genetic information passes accurately from one generation to the next, despite environmental conditions that pose constant challenges to molecular stability. Researchers still argue that the thermodynamics of biochemical reactions under extreme pressure should require enormous energy inputs to overcome the activation barriers created by crushing environmental conditions. Yet deep-sea organisms somehow power these processes with standard metabolic fuel sources while maintaining energy efficiency comparable to surface creatures operating under much more favorable conditions. The digestive enzymes produced by deep-sea creatures demonstrate remarkable activity under pressure conditions that would denature most proteins and halt normal digestive processes. These specialized catalysts break down food molecules with efficiency that rivals surface digestive systems, while operating in environments where the crushing pressure should prevent the molecular flexibility required for normal enzyme function. You observe the remarkable efficiency of waste elimination processes in deep-sea organisms, watching as cellular garbage removal systems transport toxic compounds and metabolic waste products through biological pathways that function normally despite extreme pressure conditions. These detoxification systems prevent the accumulation of harmful substances that would otherwise poison cells operating under already stressful environmental conditions. The immune system responses of deep-sea creatures involve specialized biochemical processes that continue to function effectively under crushing pressure, enabling these organisms to fight off infections and maintain cellular health despite operating in environments that would compromise immune function in surface organisms. These biological defense systems represent pressure-adapted versions of the same fundamental immune processes found throughout the living world. The hormone production and signaling systems that coordinate biological functions in deep-sea organisms operate through biochemical pathways that maintain their precision under extreme pressure conditions. These chemical communication networks enable complex behaviors, reproduction, and developmental processes to occur normally despite environmental conditions that should disrupt the delicate molecular interactions required for hormonal signaling. You notice how the blood chemistry of deep-sea creatures maintains proper pH balance and ion concentrations through buffer systems that function effectively under crushing pressure conditions. These biochemical regulation systems prevent the acidification or alkalinization that would otherwise occur when normal buffer systems fail under extreme pressure, maintaining internal chemical environments suitable for continued life processes. The specialized metabolic pathways that some deep-sea creatures use to process chemicals from hydrothermal vents represent unique biochemical adaptations found nowhere else in the biological world. These organisms have evolved enzyme systems capable of converting toxic sulfur compounds and heavy metals into usable nutrients, creating biological chemical processes that operate successfully under combined extreme pressure and chemical toxicity conditions. The circadian rhythm regulation systems in deep-sea organisms continue to function despite the absence of sunlight and the presence of crushing environmental pressure that should disrupt normal biological timing mechanisms. These internal biological clocks maintain their precision through biochemical oscillation systems that somehow remain synchronized under conditions that would throw surface biological timing systems completely out of phase. You witness now the epic story of evolutionary transformation that created these pressure-surviving masterpieces, a biological arms race spanning hundreds of millions of years, where life gradually conquered the crushing depths through incremental adaptations that seem almost impossibly precise. The fossil record of this evolutionary journey remains frustratingly incomplete, with most transitional forms lost to time, leaving scientists to piece together the story from scattered clues embedded in living creatures and ancient rock formations. The earliest ancestors of today's deep-sea creatures lived in shallow coastal waters where pressure posed no survival challenge, their bodies designed for environments with atmospheric pressure and abundant sunlight. As ocean basins deepened and sea levels changed over geological time, some populations found themselves gradually isolated in deeper waters, where survival demanded rapid adaptation to increasing pressure or certain extinction. You observe the molecular evidence of this evolutionary pressure cooker embedded within the genetic codes of modern deep-sea species, where comparisons between surface and deep-water relatives reveal the precise genetic modifications that enabled survival under crushing conditions. These genetic changes occurred through mutation and natural selection processes that somehow produced exactly the right molecular modifications needed for extreme pressure survival, a biological lottery with odds that seem astronomically against success. The timeline of deep-sea colonization reveals waves of evolutionary innovation that coincided with major geological events in Earth's history, periods when changing ocean chemistry and tectonic activity created new deep water habitats, while simultaneously eliminating shallow water refuges. These evolutionary bottlenecks forced rapid adaptation to extreme conditions, creating biological pressure tests that eliminated species unable to develop crushing depth survival strategies. The comparative anatomy of related species living at different depths shows the gradual modifications that accumulated over evolutionary time to produce modern pressure specialists. Surface-dwelling relatives possess hollow bones, gas-filled organs, and delicate tissue structures that would be instantly fatal at crushing depths, while their deep-sea cousins have evolved solid, reinforced anatomical structures that sacrifice speed and maneuverability for pressure survival. The metabolic evolution that enabled biochemical processes to function under extreme pressure required coordinated changes across hundreds of enzyme systems, each modification precisely calibrated to maintain chemical reaction efficiency under crushing conditions. The probability of such coordinated evolutionary changes occurring through random mutation seems impossibly small, yet the existence of these perfectly adapted biochemical systems proves that evolution somehow achieved this remarkable molecular engineering feat. Researchers still argue that the speed of adaptation required for successful deep-sea colonization exceeds the theoretical limits of evolutionary change through natural selection, suggesting that unknown evolutionary mechanisms may have accelerated the development of pressure survival adaptations during critical transition periods when species faced extinction from changing environmental conditions. The reproductive isolation that accompanied deep-sea colonization created unique evolutionary laboratories where pressure adaptations could develop without genetic interference from surface-dwelling populations. These isolated gene pools became biological testing grounds, where extreme pressure survival traits could accumulate and refine through generations of natural selection, operating under consistently crushing environmental conditions. You notice how convergent evolution produced remarkably similar pressure survival solutions in unrelated species that colonized deep waters independently, suggesting that the physics of extreme pressure survival severely limits the possible biological approaches to crushing depth adaptation. These evolutionary constraints channeled separate lineages toward nearly identical anatomical and biochemical solutions, despite their vastly different evolutionary starting points. The co-evolution of predator and prey relationships in deep-sea environments created additional selective pressures that shaped the development of pressure survival traits, as species needed to maintain their ecological roles while adapting to crushing environmental conditions. This dual challenge of environmental and biological pressure produced evolutionary solutions that balanced pressure survival with continued effectiveness in complex deep-sea food webs. The genetic regulatory networks that control pressure adaptation development show remarkable conservation across diverse deep-sea species, indicating that fundamental pressure survival mechanisms evolved early in deep-sea colonization history and have been maintained through hundreds of millions of years of subsequent evolutionary refinement. These ancient pressure survival toolkits provide the molecular foundation for all modern deep-sea life. The developmental biology of deep-sea creatures reveals how pressure adaptations are built into their growth processes from embryonic stages through adult maturity, with pressure tolerance increasing gradually as young organisms develop the anatomical and biochemical systems needed for survival at crushing depths. This developmental programming ensures that deep-sea creatures never experience the crushing pressure shock that would kill surface organisms transported to deep waters. The evolutionary trade-offs that accompanied deep-sea adaptation created biological specialists perfectly suited for extreme pressure survival, but completely dependent on crushing depth conditions for basic life processes. These evolutionary bargains traded versatility for extreme specialization, creating organisms that represent both evolutionary triumphs and evolutionary dead ends, perfectly adapted to their crushing environments, but incapable of surviving anywhere else. You discover now the intricate web of biological partnerships that enable deep-sea survival, relationships so sophisticated they resemble underwater cities, where different species contribute specialized services to maintain the complex infrastructure needed for life under crushing pressure. These symbiotic alliances represent evolution's most creative solutions to the multifaceted challenges posed by extreme depth environments, where no single organism could possibly possess all the adaptations necessary for independent survival. The giant tube worms you encountered earlier in your journey illustrate the most remarkable of these partnerships, their bodies serving as living hotels for billions of bacterial tenants that perform the essential service of converting toxic chemicals into usable nutrients. These bacterial partners possess the unique ability to process hydrogen sulfide and other poisonous compounds that seep from hydrothermal vents, transforming deadly chemicals into food through biological processes that would be impossible for the host worms to perform independently. You observe the intricate exchange systems that facilitate this biological cooperation, watching as the tube worm's circulatory system delivers raw chemical materials to specialized organs packed with symbiotic bacteria, while simultaneously removing the processed nutrients these microscopic partners produce. This biological assembly line operates with industrial efficiency under crushing pressure conditions that would disrupt similar cooperative processes in surface organisms. The deep-sea anglerfish presents another fascinating example of symbiotic pressure survival, where bioluminescent bacteria living within the fish's distinctive lure produce the ghostly light that attracts prey in the eternal darkness of the deep ocean. These bacterial light bulbs receive nutrients and protection from their fish host, while providing illumination services that would be energetically impossible for the anglerfish to produce through its own metabolic processes. The bacterial partners in these deep-sea symbioses possess their own remarkable pressure adaptations that complement and enhance the survival capabilities of their hosts. These microscopic organisms have evolved specialized enzyme systems that function optimally under crushing pressure conditions, while producing chemical compounds that help their hosts maintain cellular integrity and metabolic efficiency in extreme environments. You watch as cleaning stations operated by specialized deep-sea organisms provide maintenance services for larger creatures, removing parasites and dead tissue through cooperative relationships that benefit both parties, despite operating under pressure conditions that would make such delicate biological interactions impossible for surface species. These underwater service centers demonstrate how biological cooperation can overcome environmental challenges that would overwhelm individual organisms. Researchers still argue that the energy costs of maintaining complex symbiotic relationships should exceed the benefits under extreme pressure conditions. Yet these biological partnerships continue to thrive and evolve in deep sea environments, where energy resources are scarce and environmental stresses are extreme, suggesting that cooperative survival strategies provide advantages that outweigh their metabolic costs. The communication systems that coordinate these symbiotic relationships operate through chemical signals and bioluminescent displays that function effectively under crushing pressure conditions, where normal biological signaling mechanisms might fail. These molecular conversation systems enable complex behavioral coordination between species that have evolved entirely different pressure survival strategies, yet must work together for mutual benefit. The reproductive aspects of deep-sea symbiosis present additional complexity, as partner organisms must coordinate their reproductive cycles to ensure that offspring acquire the necessary bacterial partners for survival in extreme pressure environments. Some deep-sea species have evolved mechanisms for passing symbiotic bacteria directly from parents to offspring, ensuring that each new generation begins life with the microbial tools needed for pressure survival. You notice how some deep-sea ecosystems function as cooperative networks, where multiple species contribute different pressure survival adaptations to create community resilience that exceeds what any individual species could achieve independently. These biological communities represent distributed pressure survival systems where specialized adaptations are shared through complex ecological relationships. The biochemical exchanges between symbiotic partners involve specialized transport mechanisms that can function under extreme pressure conditions, moving nutrients, waste products, and chemical signals between organisms through biological pathways that maintain their precision despite crushing environmental forces. These interorganism transport systems represent some of the most sophisticated biological engineering found in deep-sea environments. The immune system modifications required for successful deep-sea symbioses involve complex adaptations that allow host organisms to distinguish between beneficial bacterial partners and potentially harmful microorganisms, while operating under pressure conditions that would compromise normal immune function. These biological security systems maintain their protective capabilities while allowing beneficial partnerships to flourish. The evolutionary history of deep-sea symbioses reveals how these partnerships developed through gradual mutual adaptation processes that occurred over millions of years, with each partner species evolving complementary pressure survival traits that enhanced the survival capabilities of their biological allies. These evolutionary partnerships represent some of the most sophisticated cooperative relationships found anywhere in the natural world. The metabolic integration between symbiotic partners reaches levels of biological cooperation that blur the boundaries between separate organisms, creating hybrid biological systems where host and partner function as unified pressure survival units that share metabolic processes, waste elimination systems, and even genetic regulatory mechanisms under crushing environmental conditions. You encounter now the ocean's most remarkable biological travelers, creatures that accomplish what seems impossible by journeying regularly between the crushing depths and surface waters without suffering the explosive decompression that would destroy most deep-sea life. These vertical migrants represent evolution's ultimate solution to pressure adaptation, organisms that have mastered the art of existing successfully in multiple pressure worlds during their daily lives. The lanternfish that begin their ascent from crushing depths each evening demonstrate biological engineering that would make aerospace decompression specialists weep with envy. As darkness falls at the surface, millions of these small deep-sea residents begin their journey upward through pressure gradients that would kill most organisms, their bodies somehow adjusting in real time to pressure changes equivalent to traveling from the deepest ocean trenches to mountain peaks in a matter of hours. You watch as these remarkable creatures navigate through the pressure transition zones that separate their crushing deep water homes from the near-surface feeding grounds they visit each night, their bodies undergoing rapid physiological changes that maintain cellular integrity despite pressure variations that would cause catastrophic biological failure in less adapted species. The speed and precision of these adaptations rivals the performance of sophisticated mechanical pressure regulation systems. The swim bladder adjustments you observed earlier reach their most impressive performance levels in these vertical migrators, where gas-filled organs must rapidly compensate for enormous pressure changes while maintaining precise buoyancy control throughout the migration journey. These biological pressure vessels demonstrate real-time volume adjustments that would challenge the capabilities of advanced submarine ballast systems operating under similar conditions. The blood chemistry modifications that enable successful pressure transitions involve rapid changes in gas solubility and protein configuration that prevent the formation of lethal gas bubbles during ascent while maintaining proper oxygen transport throughout the migration process. These biological decompression systems operate automatically through mechanisms that remain largely mysterious to human researchers studying pressure physiology. The feeding strategies that drive these remarkable vertical journeys take advantage of the abundant plankton and small fish that concentrate in surface waters each night, creating biological highways between crushing depths and food-rich shallows that support some of the ocean's most important ecological processes. These daily migrations represent the largest animal movements on Earth, yet they occur largely hidden from surface observers. Researchers still argue that the energy expenditures required for daily pressure transitions should exceed the nutritional benefits gained from surface feeding. Yet these vertical migrants continue to thrive and maintain stable populations, despite the enormous metabolic costs associated with their pressure-defying lifestyle, suggesting that evolutionary benefits outweigh the apparent energetic disadvantages. The predator avoidance strategies employed by vertical migrants involve sophisticated timing and behavioral adaptations that minimize exposure to surface predators while maximizing feeding opportunities in productive surface waters. These behavioral patterns must coordinate with the physiological processes needed for successful pressure transitions, creating complex biological schedules that balance feeding efficiency with pressure survival requirements. You observe the remarkable phenomenon of reverse vertical migration, where some deep-sea predators follow their migrating prey towards surface waters each night, creating temporary deep-sea communities in shallow environments where crushing depth specialists hunt among surface dwellers in an ecological mixing that occurs nowhere else in the ocean. The reproductive strategies of vertical migrators involve complex timing mechanisms that coordinate mating and spawning activities with pressure transition capabilities, ensuring that reproductive success occurs under optimal pressure conditions, while maintaining the migration patterns essential for feeding and survival. Some species time their reproduction to coincide with specific phases of their vertical migration cycles. The sensory adaptations that enable navigation during vertical migrations involve specialized pressure-sensing organs and internal timing mechanisms that provide accurate depth information throughout the journey between pressure worlds. These biological navigation systems guide creatures through thousands of meters of water column with precision that rivals electronic depth measurement devices. The community ecology of vertical migration creates temporary biological elevators that transport nutrients and energy between deep sea and surface ecosystems, connecting crushing depth food webs with surface productivity cycles through biological processes that play essential roles in global ocean nutrient cycling and carbon transport. The physiological recovery mechanisms that enable repeated pressure transitions involve cellular repair systems that can quickly restore any pressure-related damage while preparing biological systems for subsequent migration journeys. These recovery processes must operate efficiently under time constraints imposed by daily migration schedules that cannot be delayed without compromising feeding success. The evolutionary origins of vertical migration behavior present fascinating questions about how creatures originally adapted to crushing depths, developed the additional capability for pressure transition survival, creating biological systems sophisticated enough to thrive in multiple pressure environments while maintaining all the specialized adaptations needed for deep-sea survival during their crushing depth residence periods. As your journey through the crushing depths draws to a close, you begin to drift slowly upward through the layers of liquid darkness that have revealed their most closely guarded secrets tonight. The remarkable creatures you have encountered fade gently into the blue-black shadows around you, their impossible adaptations becoming distant memories as you rise through pressure gradients that tell the story of life's incredible resilience and creativity. Each organism you witnessed represents millions of years of evolutionary refinement, biological solutions so elegant and sophisticated that they seem almost magical in their perfection. The proteins that fold into impossible shapes under crushing pressure, the cellular membranes that maintain their delicate balance while surrounded by forces that would destroy mountains, the gas-filled organs that somehow exist where physics says they cannot. All these marvels of biological engineering continue their quiet work in the eternal darkness far below. They remind us that life finds ways to flourish in conditions we can barely imagine, creating solutions to survival challenges that push the very boundaries of what seems physically possible. As you ascend through the final pressure barriers that separate the abyss from the surface world, the crushing forces that define existence in the deep ocean gradually release their grip, like invisible hands slowly opening to allow your return to the realm of atmospheric pressure and sunlight. The creatures that call those depths home remain in their crushing paradise, their bodies perfectly adapted to conditions that would instantly destroy any surface dweller, living proof that evolution can accomplish engineering feats that surpass human imagination and technological capability. Rest now, knowing that far beneath the waves, in depths where mountains would crumble and submarines would collapse, life continues its ancient dance with forces beyond comprehension, creating beauty and wonder in the planet's most extreme environments, where the impossible becomes routine and the miraculous becomes simply another day in the deep. End of script. Sweet dreams.